Thanks, Wendy. Carolyn? And I believe you have slides that you're going to use. So.
Yes, I do, and thank you for inviting me to talk about an issue that's a rather orphan issue, but is directly related to the quality of the data that would be given back on any level for any patient, clinically or within the research setting. And it's to talk about pre-analytics. And just to put everyone on the same page, this is to show you that it's long been known that that specimen collection, handling, processing is very much related to bias in the data, the quality of the data in any form of research. And from a pathologist point of view, someone who has practiced medicine, I also was the chair of the pathology committee of the Cancer and Leukemia Group B, and so ran the biobank for a large clinical trial group for 15 years, and ran the Office of Biorepositories and Biospecimen Research at the NCI for 7 years, know the impact of the quality of biospecimens from patients on research. And how difficult it is to control that quality. So this was from a slide set from the NCI, where we saw the research is starting and ending with the patient, actually biospecimen collection, processing, and analysis. This is what we've been focusing on so far today, the data from the analysis that would go back to the patient. But in fact, the analysis data going to decision-making in clinical medicine also comes from this process, and often it is the very same specimen, as was brought up earlier, that is used for both clinical decision-making and shared for translational research. It's not separate specimens, and oftentimes the eligibility for a clinical trial is determined by the analysis of the biospecimen in the pathology laboratory. But the quality of what happens in the pre-analytic phase, everything that happens between the patient and the analysis laboratory is pre-analytic. And the quality of what happens here very much determines the quality of the analysis data and therefore its applicability to decision making. Just to use surgical tissue as an example, and of course from the NCI this was cancer specimens for the most part. The pre-analytical phase represents a lot of processes and a lot of people that touch the biospecimen, a lot of physical forces and chemical forces, antibiotics, anesthesia, etc., that affect the biology of the biospecimen, and after it comes out of the patient, what happens to it in the pathology laboratory, time of fixation, the temperature of the room, all of these things in transport and storage affect the quality of the analyte that is used in the analysis step. So the storage, even after analysis, may affect clinical and research results because it's often the stored specimen that goes back for secondary analysis later on, either in the study or in the patient's management. And just to convince you that this, in fact, does make a difference, these are published results showing two very common examples of the loss of a biomarker signal with just one pre-analytical variable, and that is time sitting on the pathology bench before the specimen is stabilized, either fixed or frozen, and the loss of signal in just 2 hours of time. So this would change the readout of a 2+ positive HERCEP test for a woman with breast cancer to a negative result. Purely spurious, having nothing to do with the biology of the specimen, or you can have a gain of signal. It depends on the specimen that and the analyte that you're dealing with. And of course, this is also true for blood specimens. We're dealing with this in the CAP-ASCO liquid biopsy group, and also the Moonshot blood pack group. All of the people and processes that represent pre-analytical steps in processing of blood samples, and these are some of the published results of different steps in pre-analytics that do make a difference, that change your analysis data results. And just to convince you that this problem will not be fixed by CLIA certification. We go to great lengths to control the quality of what we're doing in pathology, the place where the test is done, that capped in CLIA certification, and the quality of the laboratory and its operations. We control the people doing the test, education, proficiency testing, platforms that are approved by the FDA, and processes followed, SOPs and quality management. But in fact, for patient samples, it is variably varied and uncontrolled. There is no requirement right now in clinical medicine or clinical research to either control pre-analytical variables or even to record them. So for most specimens that are used in the pathology department or research, we know very little about them, and more is known about the quality of beef in a supermarket than the quality of human specimens in pathology, because the FDA regulates this. So I rest my case that this is a very important issue and needs to be addressed both on the clinical level and on the research level. And in fact, we're looking forward to a time when there won't be a separation between these things. This will be a continuous feedback loop and a learning system, and this process will feed both of those systems.
Thanks.